start by welcoming everyone to another session of Food for Sustainability webinars and reminding you that we are still talking about sustainable nutrition. Today, we invite you to join me and Margarida Palma, which is our microbiologist. She's a PhD and works here at Food for Sustainability to explore the world of biotics. And to guide us in this journey, we have the pleasure to have with us Natalia Drabińska. And Natalia, she's an associated professor at the Department of Chemistry and Biodynamics Food. And Natalia will be guiding us through all the biotics that live in our gut. Natalia, it is a pleasure to have you here. Thank you so much for your time and availability. And also with us, we have Manuela Pintado. She's the director of CBQF, which is the Chemistry and Biotechnology Center, which is a state associate laboratory. And Manuela will be showing how food waste and namely byproducts can in fact be gut allies. Manuela, it is a true pleasure to have you here with us. Thank you so much. And as I mentioned, healthy biotics are an important consequence of sustainable nutrition. And let me remind you that sustainable nutrition refers to the ability of the food systems to provide all the nutrients and energy needed for the entire population, but of course, without compromising the needs of the future generations. So it's not only about us, it's also about what we leave behind. You know me, I'm Claudia Costa. I'm the coordinator of Food for Sustainability Academy, which is the area of the Food for Sustainability where we dedicated to education, to knowledge, to dissemination, to awareness raising programs, to work towards the mission of Food for Sustainability, as you know, is to work towards the sustainable intensification of the agri-food system for a better health and well-being. So we cover topics such as circular economy, sustainable agriculture, the territory, functional nutrition, and of course, we do this all under a framework of innovation and entrepreneurship. And just to let you know that, for example, tomorrow we will have discussions around the new pack, and the new pack also here as a building block for sustainable farming. And of course, consequently, what we need to achieve is sustainable nutrition. So if you can, join us at Arruda, this time not online, but physical. We'll be there from 9 a.m. The entire morning for a very interesting discussions. Also, let me remind you that these webinars are really a team effort. They wouldn't be possible with the support of many of you that are here and others that are not here. So I would like to thank all Food for Sustainability team. I particularly acknowledge the byproduct valorization team. I have here with me Daniela Fonseca, which is our green chemistry PhD. Also Rita Silva, the microbiologist. Hello, Rita. And also Silvia Moreira, she's our food scientist PhD. Thank you so much for your support, and also to acknowledge the support from BGI for their dissemination, namely the marketing manager, Eduarda Monteiro, and BGI CEO, Gonçalo Amorim, for their relentless efforts in making these sessions possible. Before we proceed, let me remind you the house rules. Probably you already know them. You know that these webinars are recorded. So if you do not wish to appear, I just kindly ask you to keep your cameras off. You don't need to turn your cameras off, but if you wish, if you don't wish to appear, it's better to just keep your cameras off. Also, questions are welcome, and questions are very important, not only because we learn from the answers, but also we learn with the questions, with different perspectives, with different point of view. So please take part with your questions, and Margarida will pick up your questions and address them to our speakers. You know that we use Slido for this, but if you think that if you find Slido a little bit cumbersome, which probably you don't, we are all very technology savvy, But if you feel more comfortable, you just use the chat here on Teams, and we'll make sure that your question is then posted into Slido. So don't be refrain from taking part in our session. And we just mentioned Slido, right? And again, I think the team has just prepared a couple of questions for you, a short quiz on Slido. So how to go to Slido, just open a browser and just type slido.com. Once you enter Slido, you will have a hashtag box. You just insert F4S Academy and you are in. You have two tabs, one for Q&A, where you will post your questions, and another one for the polls. So let's see what the team has prepared for you today. Let's see if we continue to have a great knowledge about our topics. So the first question and relates to the gut, which we'll be addressing during this session. So do you know how many bacterial species live in our guts? So is it in the range of 200, between 300 and 500, over 500, but less than 1,000, or more than 1,000? And we are talking just about bacteria. How many of them live in our guts? And as we know, our gut is populated by different microorganisms, and the majority are bacteria and fungi. And the number of bacterial species in our gut is, you're absolutely right. It's more than 1,000 species of bacteria in our guts. So we definitely need to look after them very, very careful. And Manuela Pintado will surely highlight all the functionalities and the interactions that happen in our guts. Well done. Thank you so much. We have a second question. Do you really know what probiotics are? This is a word that, in fact, we use quite often, but do you really know what they mean? And we propose you here 4 different options. So probiotics are living microorganisms from fermented products and accurately identified. Are they dead organisms with a known origin, accurately identified, but resistant to gastric juice and bile acids, with health-promoting properties confirmed in clinical trials? And it's true that we hear a lot about clinical trials and probiotics. Or are we talking about all microorganisms that are resistant to gastric juice and bile acids? Or is it something even more complex, which are living organisms with a known origin, with accurately identified resistance to gastric juice and bile acids, but with health-promoting properties that have been confirmed by clinical trials and been confirmed to clinical trials? And so we have a winner now. And as we know, the definition of probiotics is definitely very important, because we know that marketing campaigns frequently use the word of probiotics regarding natural fermented products. And we also know that some big companies had to withdraw their marketing claims and even withdraw some products from the market. So it's also important for us as consumers to really know what probiotics are. And Natalia is going to explain better this definition, but the correct answer is living organisms with a known origin, accurately identified, resistant to gastric juice and bile acids, with health-promoting properties that have been confirmed in clinical trials. So well done. It seems that we are spot on the definition. And we have another definition for you, which will also be the topic of both Natalia and Manuela. Do you know what prebiotics are? Are we talking about probiotics that are in inactive states? are we talking about food ingredients that promote the growth and the metabolism of intestinal microorganisms? Or probiotics refer more to fermented ingredients that are good for our health? Or finally, substances derived after the microorganisms are no longer alive in our guts. So while we vote, let's see if everyone has already voted. And I wish, this audience will be doing all the national exams. Portugal will be rating very, very high in terms of academic profile. So you're absolutely right. We are talking about food ingredients that promote the growth and the metabolism of intestinal microorganisms. So well done. Thank you so much for taking part in this very short quiz. I think this was a very good appetizer to start our discussion. So thank you, team, and thank you for taking part. And I will now hand the floor to Margarida Palma, who is going now to moderate and lead this session. Thank you.
Thank you, Claudia. So it is a great pleasure to have you both with us today and to have you all here assisting to our webinars. Thanks again, Natalia and Manuela, for accepting our invitation. So our first speaker will be Natalia Drabińska. Natalia is a scientist in the field of nutrition and biotechnology. She obtained her PhD degree from the Institute of Animal Reproduction and Food Research of the Polish Academy of Sciences, where her interdisciplinary doctoral thesis focused on the use of fructooligosaccharides and inulin as a dietary component for children with celiac disease. Natalia's research is focused on better understanding the impact of nutrition on the human body, and currently Natalia works as an assistant professor of the Poznan University of Life Sciences and at the Institute of Animal Reproduction and Food Research of the Polish Academy of Sciences, where she's involved in many research projects related to metabolism, inflammation, and ketogenic diets. Natalia's presentation today is entitled Biotics, New Classification and Their Importance in Gut Barrier Integrity. So next we will have Manuela Pintado. Manuela is Associate Professor of the College of Biotechnology of the Portuguese Catholic University in Porto, Associate Director of School of Biotechnology, Director of the Chemistry and Biotechnology Center, and President of the European Biotechnology Research Institute. In research, Manuela is the head of Bioactive and Bioproducts Research Laboratory. Her main research interests include development, characterization, and validation of bioactivity of functional and bioactive ingredients, and study applications on the development of novel functional foods. Manuela's research is also focused on byproduct and new resources valorization through bioprocesses and extraction processes aiming to obtain molecules or extracts to be applied to food and other industries. So Manuela's presentation is entitled Opportunities in Sustainable Ingredients Towards Healthier Gut Microbiota. So it is a great honor to have you both in this webinar session. So I will now pass the word to Natalia. The floor is yours. You can share your slides with us.
Can you see my slides?
Yes, it's perfect.
Okay, thank you, Margarida, for a very nice introduction. As you said, today I will talk to you a bit more about biotics and their new classification and a bit about the importance of biotics in the maintenance of gut barrier integrity. Few words about microbiota. As we heard, we have more than 1,000 different species of bacteria, or only bacteria in our gut, and we have milliards of bacteria there. But not only in our gut, we have bacteria on our skin, in our lungs, and different body parts, and they are important elements of our life. It is estimated that it takes approximately 1,000 days for a child to establish the fully grown microbiota. And in the past, it was considered that our gut bacteria are approximately 2 kilograms. Now we know that it was a bit exaggerated, and our gut bacteria weighed approximately 200 grams. And each of us has its own individual unique microbiome, which contains bacteria, viruses, fungi, yeast, and so on. The microbiota is an important element of our gut, which plays very important roles. First is a metabolic role, which is including first the digestion, the supporting the digestion of food products, which normally our gut which would not be able to digest, but also our gut bacteria produce. And not only bacteria, but our microorganisms in the gut produce many products, many metabolites, which are important for our health, including vitamins, vitamin K, vitamins B, or folic acid. Then the microbiota is making a physical barrier. It's a element of our gut barrier, which is protecting our body from transport of harmful substances. It's playing a protective role as well, because also our bacteria are able to neutralize the toxins which we consume, but also toxins which are the metabolites of our other bacteria, for example, pathogenic bacteria, and immunological role, which is the stimulating of the immunological system. And this is an ideal world when our gut bacteria and our gut microbiome is balanced, but very often we can talk about dysbiosis, which is a state in which the amount of and the composition of our microbiota changes. And in the most common is when we have like not enough good bacteria, and at the same time we have too many harmful pathogenic bacteria. However, recently we also have a bit different type of dysbiosis, which occurs quite often, which is a bacterial overgrowth, which is a situation where our bacteria, instead of being in the gut, in the colon, they transfer to the small intestine. And there are very many factors which contribute to this dysbiosis, which some we can affect, some we cannot, like age. This we cannot change much. We will get older, so our microbiota will get older as well with us, but we can other factors which can affect it is the obesity, it's some pharmacological therapies, it's a lot of stress, what we eat. So the well-balanced diet will affect our microbiota, or other diseases will also, such as any inflammatory states in the gut. And we have to remember that all we eat have a great importance on our gut microbiota. So for example, just a simple example, if we have a diet rich in fiber, we will have more Prevotella bacteria. But when we will have a diet rich in fat and protein, we will have more bacteria from Bacteroides. And then we have a group of substances which will strongly affect our microbiota in a positive way. And these are the family of biotics, which include probiotics, prebiotics, symbiotics, postbiotics, and palabiotics, which are the main groups. Now we have a lot of new words arriving, psychobiotics and other, but this is the main classification based on the type of substance. So first is the probiotics, which are the microorganisms that in the right amount, which is a very important element, cause multidimensional beneficial effect for the functioning of the human body. And this also is important both local and in a systemic way. So what does it mean? It means that we have probiotics which are like super good for our gut, where they are in the real time, but also the metabolites that they are producing have a good positive effects on our body and the organs which are not located in the gut. And here we can have different probiotics depending on like kind of, let's say, the structure. So there are individual species of lactic acid bacteria. They can be yeast like Saccharomyces subspecies. We can have molds, but also the combination of bacteria and other groups like lactic acid bacteria combined with yeast. And where we can find probiotics, the real probiotics, first of all, we find in the medications and supplements, which we can find in the pharmacy, but also the probiotics are present in probiotic products like probiotic yogurt. They can be the kefir, but also the full variety of fermented products like sauerkraut, which is fermented cabbage, very popular in the central Poland, fermented cucumbers, kombucha, fermented beverages, kimchi, and all the products which are typical for different cultures. But what we have to remember, not every fermented product is probiotic, because to be classified as probiotic, bacteria needs to be alive and survive the technological process. So if we have fermented cucumbers and which will be pasteurized, it can be an option that this bacteria will not survive the process of this thermal process. And what is the second most important part is that bacteria in the product needs to be classified as probiotic, because if we have a fermented product with random bacteria, but they will not be classified as prebiotic, we cannot call this product as probiotic. And what is important, we have to always remember that each bacterial species has different biological activity, and these bacteria, which contain protein in their structure, can be allergic. So, and this not appropriate prescription of probiotics can also be harmful. Therefore, always the therapy with probiotics should be discussed with medical doctor, who should select the dose and the type and species of probiotic. The next group are prebiotics, which we can simplify as a food for good bacteria, right? Which is like which the consumption of these prebiotics can indirectly through bacteria affect positively host health. And prebiotics are resistant for digestion in the upper intestinal tract, so they are reaching the colon in the intact form. They have to reach the colon in the intact form where the microbiota will use them. And in the last years, the definition of prebiotics became a bit simplified, so it's important that it affect positively the microbiota. So in the past, it was mostly like the inulitine fructans and this kind of products. Now this group of prebiotics is much more wider, including also, for example, polyphenols. And how does it work? We consume the prebiotics, which not us, but our bacteria will consume. And then due to their fermentation activity, they will produce metabolites, and this important one, which are short-chain fatty acids, which include acetate, butyrate, and propionate, which then have positive effects on our body. First, in a local form, so they are decreasing pH in our gut, simplifying digestion and solubility of mineral complex, but also stimulating enterocytes. But also, these short-chain fatty acids can enter the bloodstream and have a positive effect on different parts of our body, including brain. And where we can find prebiotics, again, we can find in the market, like prebiotic substance, prebiotic chocolate bars or different products, but like natural sources of prebiotics are chicory, leek, asparagus, onions, cereal products, bananas, artichoke, and so on. And here it's an example of a study which was my PhD project, which was focused on the effect of prebiotics. In this case, it was inulitine fructans mixed with fructooligosaccharides on the gut barrier in a group of pediatric patients with celiac disease following a gluten-free diet. So it was a pilot study, small, on a group of 30 people, and we were before and after the supplementation for 3 months, we were checking how it affects gut barrier. And we talked recently a lot about gut barrier. In a healthy situation, we have all the cells together, and in the case of leaky gut syndrome, this the gaps between the cells cause the leakage. So the intestinal content which should stay in the intestine can reach our bloodstream, and this can cause a serious harmful effect, including inflammatory processes. And in our study, we analyzed different markers of gut barrier. And here we noticed only the difference in case of intestinal fatty acid binding protein, which is epithelial damage marker. However, we didn't observe any difference between the prebiotic and placebo group. In both group, only the effect itself of gluten-free diet was beneficial. Then we checked also the gut barrier using functional test, which is sugar absorption test. So we are comparing the absorption of 2 different sugar, one which should cross the barrier easily, which is mannitol, and then lactulose, which should not transfer, enter our bloodstream in case of the fully working barrier. And here we also didn't observe anything, like we didn't see any difference, but then we stopped and we thought, did these children actually have a leaky gut before the intervention? And then when we subgroup the group of children which had leaky gut syndrome before the intervention, we noticed that the children supplemented with prebiotics had significantly improved gut barrier integrity compared to the one fed with placebo. And going back to our classification of prebiotics, the next group is synbiotics, which is the combination of probiotics plus prebiotics. So we have a powerful tool, fully products, which we have bacteria and the dedicated products. So we have 2 different types of symbiotics action, which can be complementary. So we have probiotic dedicated to one target and prebiotic to which have separate positive effect, and we obtain 2 separate positive effects. Or we can have synergistic action. So we have a probiotic and prebiotic targeted on the same thing, and then their action is increasing in a positive way. And the use of symbiotics guarantees that these bacteria will have something to eat, making simple, which is especially important in case of situation when the diet is not fully balanced. And then it can be that, for example, we buy probiotics, but our new bacteria doesn't have anything to eat. So that's why symbiotics are more effective than prebiotics alone. And since they prolong the activity of bacteria, stimulate their development and restore the balance of disturbed microbiota. The next group are postbiotics, and how the name suggests, these are the products of the metabolism of gut bacteria, which exhibit positive effects on human health. So all the products which are this positive bacteria produce, like this short-chain fatty acid that I mentioned, the vitamins, proteins, enzymes, biosurfactants, organic acids, amino acids, all are postbiotics. And the role of postbiotics, this is exactly the role of probiotics or biotics, because these are the products which, not bacteria itself in most cases, but these metabolites are responsible for the positive effects. So they have, of course, the nutritional qualities such as vitamin K, vitamins B, but also have positive effect on the gut health, like short-chain fatty acids. They stimulate the gut microbiota, which is ongoing situation. So we have some bacteria which produce metabolites which stimulate the next bacteria. So then this microbiota eubiosis is maintained, but also they are important in our antibacterial and reduction of infections. And then the next group are parabiotics, which is quite new biotic element, which is anything that can be an alternative for the alive probiotics. So this is example, for example, of tendinized bacteria. So we have somehow thermally treated bacteria. It's because, as we thought in the past, only the alive bacteria can have positive effects. Now we know that also not only the alive, but also the cells of probiotics can have positive effects. These inactivated probiotic cells can contain different components like peptidoglycans, surface proteins, and so on, which can have beneficial effect on our health. And what is the benefit if we look at this? If we have inactivated bacteria, and this is important for the situation of like this bacterial overgrowth. So we want to have beneficial effect, but we don't want to overstimulate our microbiota to don't have to reduce the risk of all bacterial overgrowth. And parabiotics can also have application in case of the allergy, because studies showed that some probiotic bacteria can be allergic for some group of people. So if we isolate just these positive metabolites or these individual elements, this can be a solution for people suffering from allergy. And to summarize, a few messages. And so maintaining intestinal microbiota homeostasis is very important for our health. Intestinal dysbiosis is often seen in many diseases. It's very often also the pathogenesis of the many diseases. It's observed also after antibiotic therapy, in case of unbalanced diet, in case of obesity, and many other situations. We have many solutions to rebalance our gut microbiota, to switch from dysbiosis to eubiosis using the wide range of biotics. And what is the most important element? The use of dietary supplements should be always introduced after consultation with medical doctor or dietitian, because we can select not correct species or not correct doses, and we can cause more harm than the benefits. And it's always important to balance properly our diet, rich with products naturally rich in prebiotics, probiotics. And to present, I really like these slides. It's a Poznan presentation of the biotic world, where we can see exactly what is the role of each component in biotic family. And thank you very much for your attention.
Thank you, Natalia. Thank you for your excellent presentation and for giving us this overview on the world of biotics and the importance of prebiotics also on gut barrier. So I learned a lot. So maybe I'm doing something wrong because I recently bought some probiotics online. And actually, my first question is related to that. So I also did some homework, and I went to my fridge and to my yogurts and to see what kind of bacteria was in there. So I have yogurts with Lactobacillus casei, but for example, my probiotic tablets, they have different species of Lactobacillus, Bifidobacterium, and Streptococcus. So my question is, do we have scientific evidence that a specific combination of probiotics has a specific beneficial effect? For example, adding lactobacillus is beneficial in the regulation of your intestines and to avoid constipation, for example, or other mixtures of probiotics have specific health benefits?
Recently, we have results of clinical trials, which are, for example, dedicated to specific strain. That's why very often we have not only Lactobacillus casei, but then we have a code of this bacteria. So it's this specific strain has this specific activity. So we cannot consider, like, simplify it in a way that, oh, we have Lactobacillus, so all will work the same way. Not really, because we have to, the confirmed activity is dedicated to certain strains. That's why I would avoid taking probiotics without consultation, because we can imbalance we can cause dysbiosis by shuffling too much, because we will add some bacteria. Okay, we want bifidobacterium, we want lactic acid bacteria in our gut, but with specific strains, I would be more careful.
Okay, that's a very important message. Thank you. Thank you so much. So when I was doing research on yeast, I frequently came across some studies on microorganisms that were isolated, for example, from kombucha, and that could have potential to be used as probiotic. So there is still ongoing research on searching for new microorganisms that have potential to be considered as probiotics. And what do we have to do to consider a microorganism as a probiotic? What kind of tests do we need to address to consider it as a probiotic? Sorry.
It requires a confirmation in clinical trials that it actually has confirmed positive activity. Of course, we search for new. We have in modern times, we struggle with new diseases, with new inflammatory problems and new gut problems. So we are still on a stage where we need new solutions. So I think it will not stop. And since we know that microorganisms are mutating and they are like, there is a gene transfer ongoing and different things. So it can be that in new fermented products, we will find a new organism that we didn't consider previously. We also, now it's super popular to do different fermented products at home or like so on. So we can actually produce by changing the conditions in a at-home product. We can have microbiota which no one else will have. So this is quite never-ending story, in my opinion. So we will always find something new. And to confirm, we need more studies. So we should study one by one to really confirm that this is the positive activity. So we have now, for example, specified probiotics for, let's say, this gut barrier integrity. So we have the probiotic containing Akkermansia, for example, in different type of different species, very strict species, which will be beneficial for gut barrier. This one, which will be good for IBS, another for Crohn's disease. So we have now we know more, so we have some options for specific conditions.
Okay, that's good to know. So we already have some questions here in Slido. So maybe we can look at this question. So the first one is, the more diversified are the probiotics in our guts, the healthier it is. Do they need different kind of foods to grow, or a healthy diet is enough?
In general, we want like certain diversity. If it's too diverse, it's also not so good. So we have to somehow, it's not specified what diversity should be maintained, because each of us has totally different microbiota. So we are not analyzing it like, oh, well, today I have this and 1 year later I will have something different. So it's so difficult to specify exactly what is the healthy microbiota. We know some certain groups should be balanced. So I would not say that it's better if it's more diversified, but not too much. And so the second part of this question, do they need different kinds of food? The healthy diet should be enough. If we have very balanced diet full of dietary fibers, of course, if we consider it that we have no dietary problems and we don't have any elimination diets because our body requires, like a celiac disease, when we have to eliminate gluten. But in general, I would say that healthy diet should be enough to maintain the gut microbiota.
So, and do you favor natural food or supplements to increase gut microbiota, or do you not see any difference?
In case of dysbiosis confirmed, so like I have IBS problem and I have this and these symptoms, and now it is more available to do tests on microbiota. You can like order home like a set, collect your poop and send it, so you can do that. So then I would go for the pharmacological therapy, consulted with a doctor. But on daily basis, if we just want to keep our gut healthy, I would just stick to natural products, to eat from time to time fermented products, to eat from time to time yogurt, or like to keep the diet as diverse as possible, to have full range of different food for our bacteria.
Okay, so one more question. So now after I will have to pass to Professora Manuela. So what are probiotics used for, as a health complement or treat disease?
Like now it's a new term, so there is not so many even like clinical trials using parabiotics. So this is a kind of, now we are in a stage of hypothesis more. So, but I would suggest that it's more for treating diseases, will be, if it will be more popular in future, because now it's not very common practice yet.
Okay, okay. So thank you, Natalia.
Thank you
I
very
will come back
much.
to you after discussion with Manuela. But now I will give my word to Manuela. Please, can you share your slides?
So my first word is to thank COLA for Sustainability, because it's a pleasure to collaborate and especially recognize the value of these sessions to communicate science and to also inform the citizens and even to discuss scientific topics. So congratulations and thank you for also this opportunity to share some of our work. So I think Natalia already mentioned some of my concepts, but I'm going in some cases just to talk in a more a general approach of the implications of this knowledge of microbiome sometimes, but also in my case, I'll be talking about some of new ingredients that we have been producing through byproducts. And this would bring more food and more sustainable food. And it's also important to understand if these new ingredients can also be claimed with some prebiotic or gut modulation possibilities. So that's what we'll be talking now in these few minutes. So just to give you, I'm not going to detail much more because Natalia already mentioned, but talking about how can we especially use prebiotics for the modulation of microbiota, and then mainly several examples that I'll be talking today from our research group. So I think this is the message that, in fact, sometimes when I say this number, we have more bacteria in our body than, in fact, we have in the terrestrial inhabitants. That means that we are really big. So sometimes if you are dreaming and we imagine that all these bacteria is growing, it's frightening, no? But in fact, they are, most of them, they are friends. So it's an important role of these bacteria. But sometimes a very important professor in Portugal used to say, think about all genetic material they have. So we are so insignificant because it's almost 1% of our genetic material compared to the 99% of the other material. So, and this of course is material that is distributed. You can see here, I'm going just to put the pointer. So we can see here that in general, we have big groups that are more or less the same around our body, but the density in each area, it's really different. And when you talk about this gut microbiota, that in fact is very important, and you already won the question about 1,000. It's the point here, we have so many different species in our gut, but in fact, we understand and recognize that there are quite important positive markers for healthy gut microbiome. So this positive impact is really important because now we understand that the gut microbiota is not static. The gut microbiota is communicating with our different organs, and communicating, of course, cannot specific language, but this language is communication about talking about very specific molecules. And now when we start, I don't know, 20 years ago, where we could not even know part of the microbiota because molecular biology was not so known at that moment. Now, knowing all these microorganisms, we start understanding that they release different molecules. They have interaction, their own self, with our immunological system. So this means that several of chronic diseases that now we try to understand and find solutions to cure, we now understand that part of them are based on the also, not only, but also on gut microbiome. And that means that some connection axis we call communication with the brain, and we have several interaction with the brain, very clear at the moment, and even some neurodegenerative diseases has been related. Autism, anxiety, for instance, has been proven now with some base in the microbiome, but also in the lung, like asthma, some relationship, and liver, obesity, diabetes, highly related with some substances that can produce by microbiome that can be negative in some cases, but if you can prevent, we can prevent if you have good molecules produced by good bacteria. The same, even for the kidney, for instance, there are relationships with this microbiome, but especially they are really connected with the immunological system, because as you know, or if you don't know, in the gut we have a big immunological system. So all the interaction with these microorganisms and immunological systems leads to a very important communication that in certain cases can be positive or negative. But of course, this is highly influenced not only by our normal diet, as we imagine, is arriving thousands of molecules to the gut that will be influencing all these 1,000 different species, but also it's an interaction with our environment, our emotional state, and also are interacting with, of course, and especially also with the diet. And this is very important, and this will modulate the gut microbiota and the interaction with the different organs. So one of the first papers that started in 2012 show us that we can have an healthy brain function and an abnormal brain function, and is highly related with the gut microbiota. So one of the first messages saying, so part of your brain can be affected by the quality of your gut microbiota. And of course, then we start increasing, and now we understand that this is not just the brain, but several chronic diseases that I already mentioned previously. But even at brain level, we have different aspects. But this is not just related with your external factors, diet, emotional state, but also genetics. So genetics are also affecting a lot your state. So we now relate the quality of these factors, but also genetics. And sometimes even we can now already to understand and predict some of these chronic diseases, comparing the genetics and together with the gut microbiota. But when you talk about gut microbiota, we always think, okay, let's think about how can we promote it. And until several years ago, we just studied most of the ingredients in terms of cytotoxicity, genotoxicity, doses that can induce damage in our body. So all the food industry has been using different ingredients, but never until now have been analyzed in terms of gut microbiota. So now we have a new era, and there are some studies from 2021 that start demonstrating, as a review, combining different studies, that in fact several additives in our food using through a chronic consumption can lead to some problems in gut microbiota. So we can create, as Natalia already mentioned, and I was not repeating again, the dysbiosis, so the imbalance of our gut microbiota. And this can lead to different effects like obesity, diabetes, allergies, etc. So this is really a very important fact. So that's why for now we are commenting, and we know that there is the intention to have very frequent revision, because now we have an extra factor. So the chronic use of some additives is not just toxic in certain ways, like toxicity and cells, etc., but also in the gut microbiota, they can change it, and this affects us in the chronic diseases. So, but now just to introduce my topics, we have been talking about to replace the meat through a plant-based diet, and this is a trend, as you know, and there are some evidence that some vegans or vegetarian people usually has less cancer because due to the fiber, and this has been now related in certain cases with the gut microbiota. So we know that especially the plant-based diets are very rich in fiber, and this fiber can affect through different ways, but especially also affecting a balance, a good balance of the gut microbiome. So this is an example where we can see that the plant-based, for instance, are really rich in fibers. And in Portugal, for instance, we have a deficiency in fibers. In average, we should consume 25 grams, and we are consuming 17 grams. So plant-based diets, and as you know, the 5 doses a day that the nutritionists recommend as vegetable and fruits are really relevant, because we are ingesting the fibers, polyphenols, sometimes pre or prebiotics, unsaturated fatty acids, and also all the protein that is there. And we know through the studies that they are increasing in green the good bacteria, mainly bifidobacteria is very famous, lactobacillus. In fact, some of the names that Natalia was mentioning as probiotics, and now also trying to decrease what we have to the red to negative bacteria. And of course, we can increase, they can metabolize all these molecules, and they can produce, for instance, polyunsaturated fatty acids, very helpful for the health, and also they can produce some organic acids that are very, very known. We call short-chain fatty acids. These short-chain fatty acids includes acetic acid, propionic acid, butyric acid, that in fact they are now well characterized, and we understand that they have very important role in the communication between the gut and the other organs, and even specifically doing very specific role like regeneration, like happens in the gut for the regeneration membrane with butyrate. So, but also will lead, of course, to increase these benefits, will increase the gut barrier, improving it, but also reducing some negative effects we know that are quite well related with chronic diseases like intestinal inflammation, an increase in body mass, etc. So, as already mentioned, we have been working a lot with probiotics, so prebiotics, so not the bacteria, but the feed for the bacteria. And this means that this evolution of this definition has been worked a lot by Gibson since 1995. Now we understand that not only polysaccharides can be, not only a fiber or a polysaccharide can be a prebiotic, even now we can have peptides, polyphenols, different molecules can be prebiotic. So this means that they can stimulate the growth or promote favorably the metabolic activity of this bacteria. So how can we study our ingredients? So we have been looking for creating new molecules, new extracts from the different byproducts, fruits, vegetables, as I will show you now. But we need, how can we test it? So we have a new apple flower, we have a new tomato flower. How can we understand if they are doing a good role in our gut? So first we need to simulate the gastrointestinal tract. So because when I eat, different mechanisms will be affecting that flower. So I need always to simulate, and there are very specific models now, quite standard, that we call InfoGest model. Everything is optimized, so I can reproduce in a static way with this model in my lab how the digestion is affecting this flower. So, and then when I obtain the digested flour, like in my body, I'm going to apply it to the bacteria. So what we study is, or we can study with pure bacteria and we test, okay, let's see if this digested flour is making the bacteria, the good bacteria, probiotic bacteria, lactobacilli strains and bifidobacteria grow. Or even to understand if when they grow with this fiber, they can produce the short chain fatty acids and understand if you can modulate the metabolism. But now we have also a system where we use a feces model. So we collect feces from volunteers. And then with this feces, we make in the small reactors, we simulate our microbiota, and then we apply the digested ingredient. And in this way, we can understand if they produce or not throughout time of metabolism, if they produce short-chain fatty acids or other molecules. And at the same time, we can also understand, extract DNA and understand how the different groups or most important groups of bacteria from this microbiota are evolving when they are affected with our flour, digested flour. So this is how we do it, and I'm going to share with you some results. So in fact, can we obtain ingredients from byproducts and make them also replacing some of the common commercial prebiotics in the market like inulin, fruit oligosaccharides, and that's what we're doing. So this is an example of Manuela PhD program, and we were extracting, and also a project between from Superboc, where we try to extract from the yeast that is used to produce the beer. As you know, we were producing different fractions, and one fraction that is here, this is brown, it's what we call beta-glucans that are known especially when extracted from some plant biomasses. But now we would like to test if the beta-glucans or the glucans that are present in this extract, in this flour that we obtain from this brewing yeast, could be also affecting positively the microbiota. So we characterize them, we understand that they were the beta-glucans, and then we digested them and we try to test in different concentrations. Usually these ingredients are tested in a range between 1 to maximum 3%. That's what we test in the lab. And then we can see, for instance, here, then when we use different concentrations, 1, 2, 3, we can increase the growth of the probiotic bacteria. In this case, we were working with bifidobacteria, lactobacilli, and we can also see that they are metabolizing the components and producing different organic acids. Besides the normal short-chain fatty acids, they can also produce abundant lactic acid that, of course, creates a quite good low pH for the pathogens, for instance, in the gut. So some more results for bifidobacteria, and can see also that when we use the in vitro fermentation model, so we collect different volunteers, human volunteers' feces, what we can do is to show that, in fact, they are very similar in terms of our positive control, that is a fruit oligosaccharide, considered prebiotic. And we can see that the fermentation and metabolic activity is in the butyric acid very similar, in the acetic acid, in this case, even superior, as well as for propionic acid. So we confirm that we can have an equivalent of our commercial based on flour extracted or enriched beta-glucans enriched flour extracted from a byproduct. But now the fruits are really, in fact, can we use some fruits byproducts to obtain flours that can be prebiotic or at least to modulate positively the gut microbiota. And in fact, we can see here that Deborah was producing a technology. We produced different ingredients. And finally, among all these different ingredients, we have the solid fraction. That is a flour obtained either from the stem or the peel. And in fact, can this be a prebiotic? So what we were producing is different components. You can see here our flours that we obtain like a new ingredient that can be used for different applications. And we can see that this flour is rich in different components, especially lignin, hemicellulose, and cellulose with different molecules. And usually our digestion is not able to digest them very well. But when they arrive to the gut and they are metabolized by this gut microbiota, they start stimulate their growth in positive way, or even to metabolize easily and produce a very interesting molecules. And here we did the same. So we did all the digestion, and we were also using the feces model from volunteers. And we can see clearly in this case too, that comparing the control, that is inulin, that our flour was promoting more lactobacillus during 2 days than in the same for bifidobacteria. So also a mechanism that demonstrates that they can be a potential prebiotic. Of course, all these studies are just in vitro, and then we'll need to validate in human studies. The same for the melon. We have been producing different ingredients for the melon, and among the ingredients, we also have a flour for the ingredients. So Ricardo Garcia, a PhD student, was working with this flour. We also did a gastrointestinal tract. Then we applied this in the feces model, and all this was analyzed. And we can see a very interesting topic that I want to highlight. When we digest the fiber, sometimes this fiber cannot be completely digested. But what we are doing during digestion is also to open the fiber. And when we open the fiber, this fiber in plants, they are rich in polyphenols. And this is very interesting how the digestion can help to release the antioxidant activity. So the antioxidant components are released throughout the gastrointestinal tract. And we can, besides the role in the microbiota, we can create a more antioxidant effect in our gut. So this is very positive also for healthy gut. But here we can also see that there was a certain promotion more of lactobacilli similar to fouth. In the case of the bifidobacteria, there was a certain increase, but late it was decreasing, but similar to also our fruit oligosaccharides. But what is very interesting to see is that even in the case of the organic acid, the short chain fatty acids, you can see here that this size is higher. So we even could promote much more than the commercial control. Just to give you one more example, so it's not just an oil, but we also work with olive waste streams. So, you know, when we produce olive oil, we have waste streams. And here we also with Tanya, that I didn't bring the photos, sorry. Tanya produced it 2 ingredients from, so a liquid extract, just very simple, and a pulp, a solid pulp. And they were also like flours that you can use in our, so she did the same gastrointestinal tract, all analysis I already explained. And you can see that, in fact, this biomass after digestion, it could not be so promoter of bifidobacteria or lactobacillus, but they didn't affect, because sometimes some of our flours in the market or products in the market can reduce them. So there is no effect. But what we can see, there is a ratio that shows us positive effects. That is the ratio of Firmicutes group with bacteroids. And it was not only affecting, but this ratio was very similar to the fruit oligosaccharides, so also exhibiting a good effect. And the effect, especially, there was not a strong effect in the growth, but the metabolism was really affected. For instance, acetate, with our flours, it was between 20 to 45% increase compared to the control. or even butyric, that is so important, was 50 to 88% more. So this is really relevant, and you can see that some of these flowers, and this case, of course, is not just the fiber, but also even the lipids that is bringing this type, they are modulating positively this gut microbiota. And of course, even the antioxidant activity is increasing. But what we notice is also that these bacteria that is there are taking this fiber and the polyphenols that are there. And we can see that this is the beginning of the metabolism. This is the end of metabolism. So comparing the polyphenols, that are, as you know, antioxidant compounds, they could metabolize the polyphenols and they could produce new molecules that can affect positively this, the gut itself, or eventually use it to communicate and to induce different effects in our organs. So just to finish and just final remarks, like it's very important to consider that the consumption, especially with non-digestible carbohydrates, fiber in general, but now combined with some specific protein or lipids, can affect the gut microbiota strongly. And in this case, we could also prove that we can obtain food products, good food ingredients from byproducts, that we can establish a potential prebiotic effect, especially they can modulate gut microbiota and also their metabolism. And of course, as always, we always open the door and say, but we need much more studies, especially now in vivo, to demonstrate that is really, in fact, impacting the gut microbiota in vivo, but especially that are also related with other chronic diseases that we know very surely that this plant fiber and components are affecting positively different chronic diseases. So thank you so much, our group, and thank you all for listening to me during this moment.
Thank you. Thank you so much for your excellent presentation. So it is very inspiring to see so many research studies on this topic. And we hear a lot probiotics, and as far as I know, prebiotics are not that mentioned. So it's very important to know how to feed our gut microbiota to induce their growth and metabolic activity. So I have a question regarding prebiotics, but just before, let me comment on one of your slides and hear your thoughts on the topic. So I was surprised to see that so many food additives like antioxidants, colorants, and emulsifiers are now being reevaluated by EFSA due to their impact on human gut microbiota, with consequences on chronic diseases like depression, obesity, and diabetes. So do you think there is more to come? I mean, the more studies we do on the impact of food additives on our gut microbiota, the more we understand how microbiota is modulated by the food we eat and by those additives. So in your opinion, additives and processed food are still secure, or should we avoid processed food as much as we can and eat it only occasionally?
Well, foods are safe, of course. We have been working so hard in the food industry, with the food industry, and learning in the food processing that the food is safe. The food is safe based on the knowledge we have in each moment, no? And that's why we say that try to eat a diverse diet, fresh, processed, not just processed, not just fresh, or just fish, because each one, if you just consume it, just a type of food, probably they will bring a problem. So the idea is as much we diversify as better, of course, including as always the needs that the nutritionist recommend us. But this point of is still food, we'll know later much more, of course, because one point that we know now is we are starting the studies with microbiota, and these results are based on chronic consumption. So chronic consumption is like the cancer. So it takes time to understand this chronic consumption. And another thing is sometimes when we study an ingredient in the gut, we just do more acute studies. So we are not testing during several days and several doses, etc. So it's not so, there is not so much information about chronic studies of a certain ingredient. So I believe that these results are opening for also research studies and opportunity to create new models, to create more studies, and in order we can demonstrate if new ingredients and new additives in particular are changing this microbiota. But now what we know is that this information now will alert everyone. So more studies will be done in order to prove all the additives we have in the market must be controlled in chronic studies in order to understand if there is any risk. So my point here is this also is a learning process. So sometimes we are thinking about sugar. Let's take the sugar, just take the sweeteners. But now we know that the sweeteners, when you compare the studies, are not changing so much your weight. So it's probably you are taking the sweeteners with no effect, and even you are damaging the microbiota. So sometimes these results allow even the consumers, the industry, all the food system to rethink the problem and to create alternatives. So that's why we have this new concept. Clean label means to reduce the number of additives and use natural ingredients as much as possible. This, of course, is moving forward to prevent synthetic additives that are usually those that are more evidence. There is more evidence in terms of negatively modulate the gut, so the gut microbiota. So I think this is a learning process. I'd say that until what we know now, the food is always safe, and let's invest, I would say, in research to see if other additives are also affecting negatively the gut microbiota. Find more models faster to diagnose before you put them in the market is, I would say, the best solution to guarantee that we continue putting food products in the tables.
Very well. Okay. Thank you. So just before going to the questions from our participants, I would like to ask one question about your method on testing the prebiotics. So you put, for example, the pineapple flour under physical and chemical conditions mimicking our intestinal tract, right? So after this, you put those digested fractions in contact with the microbiota that you are isolated from feces, right? Yeah. From healthy patients. Okay. So is it possible to cultivate all microorganisms from gut, or some are viable but not culturable?
No, some are not cultivable. Yeah, okay. That was the problem. We are now knowing a lot about gut microbiota because 20 years ago, most of them we just knew about the microorganisms that were cultivable. But now with the molecular biology, we could start understanding that in reality, there was so many more in the gut that we could not cultivate it easily. So that's why it's not possible. And I would say that what we try to do is to collect in each moment, to bring it with healthy volunteers they were not ingesting antibiotics, for instance, and we try to multiply them because what we want to do, it's not, of course, it's not the best model because it's not in vivo, but if you have like an increasing of lactobacillus in all the volunteers, an increasing in bifidobacillus, so this will give us a guarantee. But in this case, there are also now some techniques, even some other group is working, that we have been proving that we can freeze, create like inocula that we can reproduce and they can maintain. So in these cases, eventually we can have, instead of just taking all the times, we can in some cases, to be faster and to be to screen several ingredients, we can now also to use some frozen inocula that we take from volunteers and maintain them in -80.
Okay, That's a good option. So Sylvia, can you put the questions from Slido? I have here one question that also relates to the first question. So one of our speakers asked, one of our attendees asked if there is any information available if fecal transplants can modulate the capacity of the body to increase the absorption of certain molecules or nutrients. So
it's true. It's true. So at the moment, there is some fecal transplants is already done in some hospitals. Of course, everything needs to be controlled, and in the case, just for a few diseases. There are some studies where they have been trying, especially for obesity, when you talk more about these chronic diseases, or also Crohn disease. But in this case, the obesity, because it's clear, for instance, that an obese person has a quite different gut microbiome compared to a non-obese person. So the idea was, can we change the microbiota? And it's very clear also that even this happens in animals. So the point here is there is a huge advance, okay, for certain diseases, and some groups in hospitals are already producing fecal transplants. Of course, there are still some questions about the donors. Who is the donor? The donor is taking some information that is not controlled yet. But in fact, these fecal transplants, it's a future, because if you can reduce this disease, take or prevent even, it's really important, especially treat diseases that are not easily controlled by drugs, and is taking to a huge suffering. So that's, yes, I believe that fecal transplants, but of course we are not, fecal transplants is very invasive, so we are just leaving this for now specific diseases, and as I would say that in future probably we can do it for not such critical diseases, but open more, but we need some more sure about the donors and the mechanisms, and some more information before you can generalize this type of procedure.
Okay. The other question we have is the linkage between brain and gut. Can explain somehow the gut feelings?
Yeah, I would say yes. So clearly, just to tell you, for instance, in the publications, when we have patients with Parkinson, when before they start Parkinson, the most neurological symptoms, they start, for instance, with some constipation for some period. So it's clear that there is a relation. So there are changes in the, and even when we compare this microbiota, there are differences between these patients compared to a normal person in the family, for instance, that is not sick. So this is clear. So what is happening there is that the point here is the brain controlling the gut, or the gut controlling the brain, no? Because this is always the point. So clearly is that we know that in certain diets, the metabolic activity of the microbiota, release molecules that can even affect our thinking way or our emotional state, especially depression and anxiety is now very critical and very related with the gut microbiota. And eventually even we notice that some food products can affect us also emotional state, no? But in this case, it's now clear that also some hormones we are controlling by brain are also affecting our gut. So there is this communication that we call the brain and gut axis, and that clearly will be affecting both sides. But it's proven there are molecules like GABA, GABA-aminogenic acid. We have some serotonin, even organic acid. I was telling the short chain fatty acids, propionic acid, etc. All these molecules are the communication capacity that the microbiota is doing. And these molecules are moving through the bloodstream, but also inducing our local gut entrance transferring information in a different way, like with cells, for instance. So this is this communication is true, and it's, I would say, in both ways.
Okay, thank you. Do we already have gut-friendly diets? Is it possible that they are a reality?
Sorry, I could not. Which one
Do
are
we,
you?
the second one. Do we already have gut-friendly diets? I never heard about
Yes,
gut-friendly
yes.
diets, but it's
Yeah,
a
yeah.
possibility.
At the moment, if you do your microbiota profiling, you can understand the number of bacteria that, or your dysbiosis, okay? And we can especially decide for a type of prebiotic or a beta-glucan or inulin. So because depending on the molecule or the component, we can change positively your microbiota. So if you get now, you can do your gut microbiota profile, and from there, even if you have any inflammatory bowel disease, for instance, you can do it, and even you can be recommended by the nutritionist. How can we modulate my diet in order I can positively modulate my gut microbiota? Yes.
Okay, that's amazing. I didn't know that. So we have another question. I will try to read it because it's some words are just sticked. So these results open the door to added value to add value to several byproducts. From your experience, these results will prompt industry to create nutraceuticals and enriched foods.
I would not say nutraceuticals, because a nutraceutical in the food industry is not the best solution, because a nutraceutical is a molecule or an extract that is included in a pill. So it's more a food supplement. So, but I would say that this type of flours can, on one side, be used to develop some like functional foods that can promote your health positively. So functional foods, and sometimes what is interesting is that we are needing new ingredients, because we are lacking a lot of raw materials in the world to have more food. And sometimes we can use these byproducts as a raw material, because we need more food. And at the same time, knowing more about these flours and new ingredients, we can claim for the industry, claim nutritional claims, and guarantee that we can promote the consumption of that food products. And I think this is a benefit. So, and the point here is still, some of them are already in the market, so we are not creating just everything new. A lot of them are already in the market. What we need to do with research also is to demonstrate more and more the good benefits also of these ingredients, in order that more industries can valorize more byproducts, and more ingredients can appear in the market. So I think this, but yeah, I would say that not nutraceuticals, because are more the food supplement industry working on that, but enriched foods, as you mentioned here, it's possible, yes.
Okay, thank you. So we have one more question in the chat, and after I will ask a question for both of you. So we have this question. Actually, this question was addressed previously when Natalia was answering questions from our audience. So what are the implications or risks of bacteria overgrowth? I don't know which one of you would like to reply to this question.
I can reply.
Yeah.
So the risk for bacteria overgrowth can be any problems in the kind of the structure of the intestinal tract. So if we have some gastric surgery, this is very commonly a risk for bacterial overgrowth. Also, the inappropriate diet. It's commonly like a very Western with highly processed food can increase the risk of bacterial overgrowth. And also, and for many years we were talking about this biosis only in a position that we have not enough bacteria, but now it's a lot of people struggling with bacterial overgrowth. We eat too much. This is one thing. It's very common in individuals with obesity that there is SIBO detected. For example, is small intestinal bacterial overgrowth, which is very unpleasant state where our gut is not so productive. We have a lot of bloating. The subject is very tired with malnutrition and a lot of different health problems, which are so difficult to combine with SIBO. But yes, and then it's many diseases which are connected with some intestinal inflammations can also lead to bacterial overgrowth. Also, what I mentioned, inappropriate therapy with probiotics can be a risk of bacterial overgrowth.
Okay. So just to finish, I would like to ask both of you if there is any relationship between food that is produced by organic or regenerative practices and gut microbiota. If there are any scientific evidences showing that food produced from sustainable practices have a good impact on our gut microbiota.
I don't know if
maybe we should focus on that also for future research.
Yeah.
You don't know either, Natalia? No. Any scientific work doing this relationship?
I've never heard yet about such a study, so maybe this is an idea for a future direction.
Okay. And what are the main challenges in your work in the future regarding the studies on prebiotics or on gut microbiota? What are the main challenges on the work that you do? You can go first, Natalia.
In studies that I'm conducting with the gut microbiota analysis, the biggest challenge is the mass of data, because we have more advanced equipments now. We have new generations of sequencing. We can get more information, but the analysis of it is like huge. And also we don't have this like scheme that this is a healthy microbiota and this is not. So this barrier is so fluid. So it's so difficult to judge if it's an improvement or not. We are not yet on this stage. So I think this is the biggest challenge from my perspective.
Okay, so.
Okay, just to finish, in my opinion, this type of studies of organic or not, I think it's important always to understand organic or non-organic regenerative. I think the influence can be mainly in terms of molecules that can be more relevant in one than others, positive molecules like polyphenols, etc. But on the other side is also the negative molecules. So I would say that if you have a more intensive agriculture that expose more with pesticides, etc., I would say that eventually the chronic consumption of this can surely probably affect, so that's a key point. But there are not so many studies, no study at least that I know, so relevant or conclusive about this the agriculture system and relations with microbiota. But I would say it's a good point for future. And finally, about my point is we have been using a lot in vitro models, and we need now to pass more with nutrition groups to human studies. The point here about human studies sometimes is the diversity of diets and the control of diets, that sometimes when we have a drug, it's easy to control the result or the impact. But when we have a very diverse diets for a group that we are testing this type of new ingredients with to demonstrate to get modulation, sometimes the significance cannot be so high. So this, for me, sometimes is still what we need to do now to demonstrate this more in humans and to control more the diets in order to have more significant results that demonstrate these effects and make them go to the market with health or nutritional claims. Thank you.
Okay, thank you so much. So thank you for being here with us today. For me, it was a great pleasure to know about your work and to discuss these topics with you. I will now pass the word to Claudia that will close the session.
Okay, thank you, Margarida. Just a final word, and also it was a pleasure to be guided here to this fascinating topic and just to understand that so much of our health and of our condition, it's really actually in the guts and not in other parts of our body. It was a very interesting conversation. Also to let know our participants that Food for Sustainability Academy will start to be on the road now. So we're going to promote physical events. So currently we're going to stop these online sessions, but this doesn't mean that we are done. We will be back and we'll be posting everything in the usual means, in our newsletter, in our website, through the social media, and we'll be back for more topics. And if you want to join us in the physical events, it will be a pleasure. Natalia is probably a little bit away from your
Yes.
Natalia's. It's in Poland, but anyone who can join, and eventually, if sometimes we have some links, you're more than welcome. So thank you very much. We'll be back, and hope you have enjoyed this couple of sessions from Food for Sustainability webinars. So be in touch. Thank you, and bye.
Thank you very much.
Thank you,
Bye-bye,
Natalia.
everyone.
Thank you. Bye-bye. Bye-bye.
Bye.
Bye-bye. Bye. It was a pleasure. Nice to see you all.